Good morning, everybody, or afternoon or evening, whenever you're watching this. This is going to be first lecture on the brain and spinal cord that I have for you. And so where we're going to start is the formation of the brain and spinal cord in the embryo. So you see here, this is a picture of the embryo. We're taking a cross section through it and taking a look at it. And what we're going to notice is that there are three layers of cells. There's an innermost layer called the endoderm, a middle layer called the mesoderm, and an outer layer called the ectoderm. And what happens during the course of development is that a groove forms down the middle of the outermost layer, the ectoderm here. So the cells kind of fold in and pinch in, and then eventually those cells that have folded in roll up into a tube that is called the neural tube right here that you see. And that neural tube is what develops into the brain and the spinal cord. If you remember from our integumentary system unit, we learned that one of the vitamins that is essential for proper development of the neural tube is folic acid or folate. And that's why we believe that people closer to the equator were selected for having darker skin, because that they have more melanin that protects their folate supply, so that the neural tube of the baby develops properly, so that the baby is able to then reach reproductive age and pass down their genes. If that neural tube doesn't form properly, the child's going to have problems with either their brain or spinal cord, probably not reach reproductive age or be able to reproduce, and won't pass down their genes. So when we talk about the neural tube, like I said, three layers of cells appear. The endoderm is the innermost layer, and that's going to become the lining of the body cavities and the digestive system, which makes sense. The mesoderm is the middle layer of cells that will develop into muscles and bones, which again, that's kind of in the middle of our body, so that makes sense. And the ectoderm is that outermost layer of cells that's going to become the nervous system and the integumentary system. And like I said, a groove forms down the middle of the back in the ectoderm that rolls up into a tube called the neural tube, and the neural tube becomes the brain and spinal cord. And like I mentioned before, folate or folic acid is what's required for the proper formation of the neural tube. So I'm going to keep going. You can always pause the video to fill in your notes if you need to. I'm just going to keep going here though. So if the neural tube doesn't roll up properly, the baby can develop a disorder called spina bifida. There's three different types of spina bifida. There's spina bifida occulta, spina bifida meningocele, and spina bifida myelomeningocele. And we can see here with spina bifida occulta, the vertebrae don't completely enclose the spinal cord near the bottom of the spinal cord, but nothing actually from the spinal cord protrudes out into that gap. I found out in 2019 when I had my first bout with sciatica, and the orthopedic doctor took an x-ray of me, that I actually have spina bifida occulta, which most people don't even know they have. It's called hidden spina bifida. And had I not had to get an x-ray, I never would have known it either. I thought it was kind of ironic, actually, that I've been teaching about it for several years, not knowing that I actually had it. Okay, so the second type of spina bifida is spina bifida meningocele. And with that, the meninges, which are the coverings, the connective tissue coverings that wrap around the spinal cord, protrude out into this gap where the vertebrae didn't close completely around the spinal cord, and some of the cerebrospinal fluid will as well, but none of the spinal nerves do. So again, there may be some problems associated with this, but they're not going to be as severe as the third type here, spina bifida myelomeningocele, where not only do the wrappings called meninges of the spinal cord protrude out into this opening, not only does the cerebrospinal fluid go into this gap, but so do the spinal nerves. This causes severe neurological problems. It can cause severe pain. You're going to have people with this who are going to have real trouble walking. They're going to need to use braces if they can at all. And if they can't, they'll be in a wheelchair. So spina bifida occulta is that hidden spina bifida where the vertebrae don't completely close around the spinal cord, but the cord itself is normal. In spina bifida meningocele, we get that fluid-filled cyst that forms. The meninges that cover the brain and spinal cord protrude through the vertebrae, but there may be no complications. But with spina bifida myelomeningocele, we get a fluid-filled cyst that forms. The meninges and spinal nerves protrude into that cyst, and there are severe neurological problems with it. Prenatal surgery can be done if the doctor detects the spina bifida then that can help alleviate some of the pain associated with spina bifida, but the people will still have neurological problems. Thankfully, the life expectancy of someone with spina bifida is not reduced. So here's just another picture showing you where we have this opening in the vertebrae that the meninges, the spinal fluid, and the spinal parts of the spinal cord all protrude out into that gap that forms there. And this is what we'll see in the back where that's located. Now, prenatal surgery means surgery that's done before the baby is born. So it's done in the mother's uterus. So the mother's uterus would have to be cut open. And then the surgery is done on the baby's back to try and fix some of that problem here. Okay, so here's a picture showing you the mother's uterus for prenatal surgery, cut open. Here's that fluid-filled cyst with spinal nerves that are protruding out into it. And here is the closed up surgery. So I mentioned the meninges before. Those are the wrappings around the brain and spinal cord. So we're going to run through those real quickly here. So this is a cross section through the brain. It's a section like this that would cut the body into an anterior and posterior half, right? And so we remember that that is called a frontal section or coronal section. So what we're looking at here, this is the top of the head. This is the scalp right here. This is the adipose tissue underneath it. This is the skull bone right here. So now we're getting down into where the brain is located, into the cranial cavity. And wrapping around the brain and spinal cord, the outermost layer of connective tissue that wraps around everything and helps protect it is a really tough covering called the dura mater. Deep to that, there's a little gap, and then the next layer, the next wrapping called a meninge, actually meninx is the singular, meninges is plural, is called the arachnoid layer or arachnoid mater. And that's this layer here that wraps around the brain and protects it. We'll notice that there's a lot of blood vessels running through this space between the arachnoid mater or arachnoid layer, and then the deepest wrapping around the brain that wraps directly onto the brain and goes down into every groove and up over every hill, and that is called the pia mater. So as we look at those three meninges, the dura mater, the arachnoid mater or arachnoid layer, and then the pia mater. Those are the three meninges, okay? And again, you can pause the video here if you need to. I'm going to keep going. So the dura mater literally means tough mother, okay? And it's like a thick cap that covers and goes around the brain. And if we ever get back into the school building, we actually have a human brain in a jar that I can show you, and you'll be able to actually see this dura mater, that thick, tough covering. This encloses the entire central nervous system, so brain and spinal cord. It's directly attached to the bones of the cranium and then the end of the vertebral column. There's a space between the dura mater and the arachnoid mater that is called the subdural space. And if you get an acute trauma to the brain, like in a car accident where your head gets slammed against something, or if you get hit with a baseball bat or something, you can get what's called a subdural hematoma. And with that, blood fills the subdural space, that space between the dura mater and the arachnoid layer. And because the cranium, the skull bone can't expand, right? The only place for the blood that's filling that space to go is down, and it pushes down on the brain tissue. And that leads to brain injury. And if it pushes down hard enough, it can actually push part of the brain down out of the cranial cavity. And then that would be a hernia, and that's going to end up leading to death. The way that you would treat a subdural hematoma is to drill small holes into the cranium or skull to allow the blood to drain, put in kind of a stent or something to allow the blood to drain out and relieve that pressure, right? So a subdural hematoma would be blood filling this space right here between the dura mater and the arachnoid mater. Okay, now the arachnoid mater or arachnoid layer is the middle layer of our meninges. There's a space between the arachnoid layer and pia mater called the subarachnoid space that is filled with cerebrospinal fluid. And our bodies need to replace that cerebrospinal fluid because it fills up with waste from the cells and gets kind of dirty. And so our bodies are constantly filtering out the old cerebrospinal fluid and putting in new cerebrospinal fluid. We need to change that, just like you need to change your car's oil because your car's oil gets filled up with all kind of metal shavings and filings and all kinds of gunk from your car, And so you need to replace that oil every 3 months. Now your cerebrospinal fluid needs to be replaced more frequently, but it's the same kind of idea there. And there are those blood vessels running through the arachnoid mater that drain that cerebrospinal fluid, and then your ependymal cells will produce new cerebrospinal fluid by filtering your blood and filtering out just some of the fluid to create the cerebrospinal fluid. So here's the arachnoid layer or arachnoid mater. Here are the blood vessels running through it that are going to be absorbing the old cerebrospinal fluid, as the ependymal cells will be creating new cerebrospinal fluid. So the last layer of the meninges is the pia mater, which literally means tender mother. And that is a thin membrane that covers every convolution. That means every little groove, every little hill, every valley, every little part of the brain is covered with this pia mater. So it lines the sulci, which are the grooves of the cerebrum and cerebellum. And then a thin filament of the pia mater actually extends at the end of the spinal cord in the upper lumbar region of your back, okay, to the sacral end of the vertebral columns, to the very bottom of our vertebral column. What's interesting is the spinal cord actually doesn't extend into the lower lumbar region. It ends at the upper lumbar region of our vertebral column. So why that's important? Doctors, if they need to, can insert a needle into the lower lumbar region through the dura mater and through the arachnoid layer to withdraw cerebrospinal fluid, and that's called a lumbar puncture, without risking damaging your spinal tissue, because the spinal cord ends up towards the top of your lumbar region. So if you put a needle into the bottom part of your lumbar region, then the doctor can withdraw cerebrospinal fluid without doing any damage to your spinal cord. And they would do a lumbar puncture to look for neuropathology. So they might be looking to see if you've got some sort of infection, for example, and they can look at the biochemical functioning of your nervous tissue. Okay, so we were just looking at the pia mater there, that wraps tightly around the brain and spinal cord. And then a thin filament of the pia mater extends all the way down to the bottom of our vertebral column right here, even though the spinal cord ends right here near the top of the lumbar region of our vertebral column. And down here is where a doctor could stick a needle into this column right here and withdraw some of the cerebrospinal fluid without doing any damage to the spinal cord. Okay, so that was the meninges, the coverings of the brain and spinal cord. Now we're going to start looking more closely at the brain. We're going to start with the 4 regions of the brain. Okay, and so we've got the cerebrum, we've got the diencephalon, which consists of the pineal gland, thalamus, and hypothalamus, the brainstem, which consists of the midbrain, pons, and medulla oblongata, and then the cerebellum. Okay, we're going to focus on the cerebral hemispheres here today. We'll cover these other parts in another lecture. Okay, so here are the 4 big regions of the brain. We've got the cerebrum up here, the diencephalon here, the brainstem, and then the cerebellum. Okay, so the cerebrum makes up most of the mass of the brain, and the wrinkled part of the cerebrum, which is the outermost part of the cerebrum, is the cerebral cortex. The brain is separated into right and left hemispheres by a deep groove called the longitudinal fissure. And we actually have what's called contralateral control over our body, which means that the left side of the brain controls the right side of the body, and the right hemisphere of the brain controls the left side of the body. Okay, so what's kind of fun, which this is one thing I miss about being in class, is if you try to get your right hand to go clockwise and your left foot to go counterclockwise in the opposite direction, you can do it, and you can do it smoothly. But if you try to get your right hand to move clockwise and your right foot to move in the opposite direction, to move counterclockwise, you can't do it smoothly. It'll be all jerky. And that's because when you are using opposite hand, opposite foot, those are controlled by 2 opposite sides of the brain. But when you're using a hand and foot on the same side of the body, you've got the same side of the brain trying to send different signals to different body parts, and it does not work. Okay, there is a structure called the corpus callosum that provides the pathway for communication between the 2 hemispheres. Okay, and in severe cases of epilepsy, where patients have seizures frequently, One radical surgery that's done is to sever that corpus callosum. That kind of shuts down the electrical storm that happens in your brain when you have an epileptic seizure. People with this actually end up with a disorder called split brain disorder. And I'm going to share this video in Schoology, this link right here. If you just watch like the first 4 minutes of it, you'll see what split brain is. And it's really bizarre, really kind of cool if you're into brain stuff. So I'll share that link so you can do that. All right, so let's look at some of these structures here. The longitudinal fissure is that deep groove that runs between the two hemispheres here. And you can see this one actually looks like it's got a coating on it. And that's because the meninges haven't been removed from this hemisphere, this left hemisphere right here. The cerebral cortex is this wrinkled outer portion right here. It's made up of gray matter, so cell bodies and dendrites. And then all this down here, you see all this white matter, those would have the myelinated axons of sensory and motor neurons. So the corpus callosum is this structure right here that connects the left and right hemispheres. You can see in this section, we took a frontal section. This section is a sagittal section. Here's the corpus callosum in this brain. The cerebrum now is responsible for memory, emotion, and consciousness. The cortex is the outer covering of gray matter made up of cell bodies and dendrites. And there's a few parts to this cerebrum that we need to be aware of. And in the cortex, we have what's called the limbic cortex, which is part of the limbic system and is responsible for and involved in memories and emotions and behaviors. There are structures beneath the cerebral cortex or deep to the cerebral cortex that we call subcortical structures. There are basal nuclei that help plan movement. If you remember, nuclei are groupings of cell bodies within the central nervous system, okay? There's the basal forebrain that is deep to the cerebral cortex where some learning and memory occurs. There's the amygdala, which is responsible for emotional responses, and then the hippocampus, which is responsible for long-term memory. If you want to spend more time learning about all these things and how they are involved in all these different activities, I'd really recommend that you take psych. We just don't have time to go into all of that in this class with all of the anatomy and physiology we have to cover. So here's where the limbic cortex is located, right here. Remember, that helps to control our mood. Here is the, and here you see, here's the hippocampus involved in memory, and here's the amygdala involved in the emotions like fear and anxiety. Okay, and then finally, the basal nuclei are these structures here that notice they are deep to the cortex right here that help us plan movement. These are groupings of cell bodies in the cerebrum that help us to plan movement. Okay, here's the basal forebrain where that's located. And again, here we're going to see the hippocampus and the amygdala that are, and then the cerebral cortex is that outer layer. One thing to know, the grooves, are individual grooves in the brain are called is called a sulcus. If we're talking about all of them together, we'd say sulci. And then the hills are called gyrus, and multiple ones we call gyri. I've also heard it pronounced gyri, just so you know. So why do you think we have all these folds in the cortex? Why not just have it flat and smooth? Because the size of our heads is limited by the size of the birth canal that we have to pass through as a baby. So the folding allows there to be more gray matter to fit inside of our cranial cavity. If we didn't have all those folds, our cerebral cortex would cover about 1 square meter. Our head would be gigantic. We'd be like a huge bobblehead everywhere, and it would be impossible to fit through the birth canal. Okay. So the last topic for the day are going to be the lobes of the brain. There are 4 lobes of the brain that you need to be familiar with, okay? So there's 4 regions, remember, right? We had the cerebral hemispheres, the diencephalon, the brainstem, and the cerebellum. Now, when we look at the cerebrum, we're going to find that there are 4 lobes. There's the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. Okay, so here are those 4 lobes, FPOT, right? Frontal, parietal, occipital, temporal. The frontal lobe and parietal lobe are separated by a deep groove that runs around the brain here called the central sulcus. The parietal lobe and occipital lobe are separated by another groove called the parieto-occipital sulcus. And then we see that the frontal and temporal lobes are separated here by this deep groove called the lateral fissure, okay? So the frontal lobe has primarily a motor function. Just anterior to that central sulcus is a structure called the precentral gyrus. It's a hill just in front of the central sulcus. It's also called the primary motor cortex. So the cells in this area are upper motor neurons that tell our skeletal muscles what to do, right? So they send signals down through the spinal cord to our skeletal muscle to tell us to move. Anterior to the precentral gyrus is the premotor area where we contemplate what kind of and figure out what kind of movements need to be made. We're also going to see a very important area called Broca's area on the left side of the brain that is responsible for the production of language. So it controls the movements that are responsible for making speech, right? If you get damage to Broca's area because there's a stroke that kills brain tissue in that area, or cancer, or you get a tumor that damages that area, or some kind of trauma, you can end up with Broca's aphasia, where you can understand what people are saying to you, but you can't produce speech that conveys what you're thinking. You end up using very simple words like thing and great, and it's really hard to communicate with other people. And so we'll take a look at what happens if you've got Broca's aphasia with your speech. And then we have in the very anterior portion, we have what's called the prefrontal lobe of our frontal lobe. And that's where our personality and our short-term memory and our consciousness are controlled. Okay, so here's our primary motor cortex or precentral gyrus. Here is the premotor cortex where we plan our movements. Here's the prefrontal lobes where we have our emotions and personality and consciousness controlled. And then here's that central sulcus that separates the frontal lobe from the parietal lobe. The parietal lobe is responsible for somatosensation, which is just basically general sensations associated with the body. And that is controlled in the postcentral gyrus, which is a hill just posterior to that central sulcus. So this is where your tactile senses are processed: touch, tickle, pressure, pain, itch, vibration, where your body position in space, that's called proprioception, understanding where you are in space, and also kinesthesia, your movements. All those are processed in that postcentral gyrus or primary somatosensory cortex. So as we look at the brain here, here's our central sulcus, frontal lobe, and all of its stuff up here. This is your parietal lobe in purple, and this dark purple region right here shows you where that postcentral gyrus or primary somatosensory cortex is located. All right, the occipital lobe makes sense of visual information for us. That is the key thing right there, is associate occipital lobe in the back of the brain right there with vision. And damage to that can lead to visual confusion or even blindness. And I had a former student who went up for a dunk in basketball. He's like 6'6", 6'7", he was telling me, and somebody cut his legs out from underneath him. He fell backwards, banged his head on the playing surface really hard, and for a while there he couldn't see anything. He was kind of like blinded for a little while because of the jarring hit to the occipital lobe. All right, which you can see is located back here. And then finally, the temporal lobe, you want to think auditory information. That makes sense because it's located kind of right by the ear right here. So this processes auditory information, which is sounds that are coming in. It's important in hearing and selective listening. And no, selective listening is not what I do with my wife, where I pay attention to some things she tells me and ignore others. That is figuring out what to focus on with all the auditory information that's coming in. Are you going to be focusing on the fan that's blowing in the classroom or your teacher's voice, right? That's what happens here in the temporal lobe. The temporal lobe also allows for comprehension of speech in Wernicke's area, usually also on the left side of the brain. And that contains neurons that are involved in understanding speech. So if you get damage to that area of the brain because of a tumor or because of a stroke or some kind of trauma to the brain, you end up with what's called Wernicke's aphasia, where you don't understand what is being spoken to you. So you can speak fluently, but you're just speaking gibberish, right? Words that don't make sense together, okay? And so this just shows you, here's Broca's area in the frontal lobe on the left side. Here's Wernicke's area in the temporal lobe on the left side. And that is where I'm going to stop for today. I'm going to put a couple of videos in the update for Schoology. I'm going to put a Broca's aphasia video, a Wernicke's aphasia video, and then I'm going to show that, put that video of split brain in today's page in Schoology also. So I hope that was helpful, and I will talk to you guys after Thanksgiving.